I'm doing my master's. So today I'll be talking about my project titled Substrate Stiffness Directed Differentiation of Induced Pluripotent Stem Cells to Cardiomyocyte Subtypes. It's a long title, but we'll get to it. Okay, this is a video that I took 2 weeks ago in my lab. So spontaneously contracting cluster of cells called cardiomyocytes, essentially muscle cells, heart muscle cells. I added the sound for theatrics, not in there. Yeah, so before we get into all these cool stuff, let me talk a little bit about the motivation and background for the project. So first up, approximately 1.3 million Canadians are currently living with a damaged heart condition known as cardiomyopathy. And of that, 70,000 heart attacks occur every year. The majority of these heart attacks are non-fatal in nature, meaning that they don't cause the death of the patient, but they do cause cell death in the heart, which reduces cardiac function of the heart. Right now, there's no known cure for a damaged heart condition, unless you get a heart transplant, which we all know is hard finding a matching donor. So there's definitely a need to provide viable treatment options to restore the cardiac function of patients. So this led me to the question of how do we mend broken hearts? So right now, the leading candidate for the treatment of cardiomyopathies lies in tissue engineering and regenerative medicine using the patient's own stem cells. So these stem cells can be derived from other cell types such as fibroblasts by inducing them to a stem-like state of pluripotency. This has been done in 2007 by 2 researchers, Takahashi and Yamanaka, by using 4 transcription factors, OX4, SOX2, KLF4, and MYC. What they did is, using these 4 transcription factors, they were able to transform human fibroblasts to iPSCs, or induced pluripotent stem cells. So iPSCs, like any other stem cells, they exhibit features of typical features of stem cells in that they are 1, pluripotent, meaning that they are capable of becoming any cell type in the human body, and 2, they exhibit a feature of cell renewal, meaning that they can proliferate indefinitely given right conditions. So a separate group of researchers led by Laflamme successfully managed to transform or differentiate a monolayer of iPS cells to a spontaneously contracting cluster cell known as cardiomyocytes by using 2 biochemical agents, activin A and BMP4, bone morphogenetic protein 4. Both activin A and BMP4 are part of the transforming growth factor beta signaling protein superfamily. So in this spontaneously contracting cluster of cells, they are made up of, they are likely to be made up of consisting of a heterogeneous population of cells consisting of the following cell types: fibroblasts, nodal myocytes, atrial myocytes, and ventricular myocytes. So both atrial and ventricular myocytes are known as working myocytes, and nodal myocytes are essentially pacemaker cells that makes up the sinoatrial node, or the SA node of the heart. So that's all really nice, but so what's the problem here? So a lot of these traditional differentiation protocols primarily focuses on trying to recreate the biochemical signaling events associated with cardiogenesis, meaning to say that, and oftentimes they neglect physical parameters and stimulus such as substrate stiffness or mechanical strain. Yeah, so this has resulted in poor differentiation outcomes, especially in directing stem cell specification to specialized cardiomyocyte subtypes, so i.e. working cells versus nodal cells. So why is it important to differentiate between working and nodal cells? Well, in order to have an eventual clinical application for cell-based therapies, working cells can be used for myocardial infarction repairs, and nodal cells can be used as biological pacemakers. So we need to differentiate them between themselves. So a little bit about what has been, so as alluded to in the previous slides, there is a significant or measurable effect of physical stimulus on stem cell differentiation and growth. So for example, in a study done a couple of years back by Engler, he showed that you can direct mesenchymal stem cells differentiation to neuronal myoblastic meaning muscle, osteoblastic meaning bone, lineages by simply varying the stiffness of the substrate on which these stem cells grew. So for example, for the stem cells that were growing on very soft substrates, they tend towards a neuronal lineage, which is becoming more like neurons. And for those that are growing on an intermediate stiffness substrate, they tend towards a myoblastic lineage, a muscle type cell. And for those that were growing on very stiff substrates, they tend towards an osteoblastic lineage, so bone-like cells. Similar findings has also been reported with iPSCs, induced pluripotent stem cells. So with accumulating evidence in literature suggesting that physical parameters and stimulus having a considerable and measurable effect on stem cell differentiation, we propose that by manipulating the stiffness of substrate on which stem cells grow, it will be possible to generate either enriched nodal working type myocytes or nodal myocytes. So essentially what I'm saying is that by coupling traditional soluble factors with and modulating the stiffness of the substrate on which stem cells grow, we can improve differentiation outcomes, in particular directing stem cells to the different cardiomyocyte subtypes. So to test our hypothesis, we need a way to vary the stiffness of the substrate on which the stem cells grow. So according to a protocol set up by Francesco, this can be done by blending two commercially available polyethylene methyl siloxane PDMS subtypes, Silgard 527 and Silgard 184. And you can get a substrate of varying Young's modulus between a soft 5 kilopascal to a fairly stiff 1.72 megapascals. So there is a little sketch that I drew of how this process is. I won't go through it for time consideration, but by just simply varying the ratio of these two PDMS subtypes, we can get substrates of varying stiffnesses. So at the end, we will have something that looks like this for us. We have a Petri dish, a plastic, regular, typical Petri dish, and the PDMS substrate is sitting on top of it, and the cells sit on top of the PDMS substrate. So after characterizing the stiffness, we have the following stiffnesses for our substrates. So we have three substrates. The soft is very soft, as reported 5 kilopascal. The intermediate substrate has a stiffness of 830 kilopascals. The hard substrate has a stiffness of Young's modulus 1.72 megapascals. So just to give you a feel of what the stiffnesses are like in the natural human body, muscle tissue is approximately 10 kilopascals. Cardiac muscle under physiological strain has a Young's modulus between 30 to 400 kilopascals. Arterial walls, 800 kilopascals. So a very interesting thing to note is that all tissue culture work right now being done in the lab is on tissue culture dishes that are made of typically polystyrene, and it's very stiff. It has a Young's modulus range between 3,500 to 4,000 megapascals, so that's very stiff. So yeah, it looks something like this. You can't really see the substrate by sitting at the bottom of the well. It's transparent, so it's convenient. You can look at it under the microscope. So we initiated differentiation according to the protocol set out by Laflamme, as mentioned earlier, using the two biochemical agents, actinin D and PDE4. Now, instead of a regular tissue culture dish, we're doing it on substrates of different stiffnesses, soft, intermediate, and hard. So to characterize the cardiacness of the resulting cells, we use protein markers, alpha-actinin, cardiac troponin T, and HCM4. So essentially what I'm saying here is that by probing for this particular proteins, we can determine if they are becoming more of like working cells or becoming more nodal. case of those of you who are curious, alpha-actinin is an actin binding protein that's localized at the Z disc. CTMP, or troponin T, is part of the troponin complex. It binds to tropomyosin that helps bind to actin. And HCM4 is an intramembrane protein that is expressed in the SA node, or the pacemaker region of human hearts. So the results we got is, as shown, it's fairly interesting. So there is a trend that suggests that for stem cells that are growing on stiffer substrates, they tend towards a more nodal type lineage, meaning that there are a higher proportion of stem cells becoming nodal cells, given the same soluble condition. And the reverse is true. The softer a substrate, the more working cells we see. So this is done using immunostaining of the protein, using an instrument known as the flow cytometer. So a quick wrap up. The observations that we have right now is that substrate stiffness thus you regulate the ratio of nodal to working type cells. More specifically, softer substrate tend to enrich working type cells, and stiffer substrate tend to enrich nodal cells. So a possible explanation for this is that in the natural human heart, the SA node is actually a fairly stiff cigar-like shaped node that is surrounded by very stiff connective tissue, as shown in this figure. So this is a meson trichromatin section of an SA node. The SA node is highlighted, so can't really see, but it's over there, and it's embedded in stiff connective tissue. So what I'm suggesting is that because of the fact that you're recreating the stiffness of the SA node, we are seeing more differentiation towards a nodal type characteristic. So a future direction for this project would be to test this further and to see if we can control one step more, better control this differentiation of cardiomyocytes. So it would be interesting to see what if we grew this stem cells on soft substrates with hard islands. And since we're expecting more nodal cells to be generated on hard substrates, or hard substrates, we'd be expecting to see more nodal cells on these hard islands. And if that's true, it would definitely be something interesting moving forward. Yeah. With that, I'd like to thank my funding and some people.
Anyone with questions? Nancy?
I'm curious, what fraction of the cells at the end are the desired cell type, like the stiffer ones, what fraction of them are the nodal cells?
It's a very good question. So, give me a second. What fraction? So, how we got that those data is that we do something called immunostaining. So we stain for probe for the specific protein, and we run it through a flow cytometer, which basically counts how many times this protein is being expressed. So with that, we have a density plot, which basically counts this, and we got something like on the soft substrate, we got a really low proportion of nodal cells, less than 1%. So it's like a 0.8%. Yeah, I have the plots here, but my computer froze. Yeah.
So what is the most abundant type? Like what fraction of your cells are only one type?
Yeah, so it's not like all the cells are becoming nodal. So there's only a proportion of cells that are becoming nodal. So on the soft ones, we got a 0.8% of the cells becoming nodal, and on the stiffer ones, we got a 7% of cells becoming nodal. Yeah. Thanks for this is a very good question. Yes.
These bigger cells are excitable. Is it also possible to electrically stimulate iPSCs to encourage differentiation?
Yeah, that's a very good question. So like.